Thank you everyone for joining us on today's webinar. My name is Tom Ewing, and I am with RTI International's Forensic Technology Center of Excellence. Before we get started, let me go over how you can view and interact with today's webinar. First thing I would like to draw your attention to is our chat, which is located on the left-hand side of your screen, marked chat. This is going to be the best way to communicate with us if you have any questions regarding today's material. Feel free to use the chat to ask any topical questions you have throughout the presentation. All questions pertaining to the material will be retained until the Q&A session that will be held at the end of the webinar. This chat is also the best way to let us know if you experience any technical issues. If you do experience any issues, please feel free to type them into the chat, and either myself or one of our customer service representatives will make sure you will get taken care of. In order to have the best viewing and listening experience, we recommend using the latest versions of the following internet browsers: Microsoft Edge, Google Chrome, Apple Safari, or Mozilla Firefox. All your audio for today's webinar is going to be streaming through your computer speakers. If for some reason you happen to lose your live audio or live video feed, the first thing we would like you to do is simply refresh your web browser and then hit the play button in the middle of your live feed window. This should refresh your connection with both the live audio and video feed for today's event. If for some reason this does not work, or you experience any other technical issues, please make sure to let us know in the chat pod. You can also utilize the toll-free number and our email address that are located directly above the live feed window to contact us with any customer service issues. We always like to find out a little more about our audience, so if you could take a few moments to type in your location into the chat, this will give us some great feedback about where everyone's joining from and ensure that your chat function is working properly. Today's webinar is slightly different than our normal webinar. We have four amazing presenters here today to give their insights into emerging methods for body fluid analysis. In addition to our unique format for today, we also have a special introduction from NIJ Senior Physical Scientist Chuck Hurick. Without further delay, here's Chuck to introduce today's topic and presenters.
Hello, my name is Chuck Hurick, and I'm a Senior Physical Scientist with the National Institute of Justice, or NIJ. NIJ is the research, development, and evaluation agency of the U.S. Department of Justice and is dedicated to improving knowledge and understanding of crime and justice issues through science. NIJ provides objective and independent knowledge and tools to inform the decision-making of the criminal justice community to reduce crime and advance justice, particularly at the state and local levels. NIJ's pursuit of this mission is guided by the following principles. Research can make a difference in individual lives, in the safety of communities, and in creating a more effective and fair justice system. Government-funded research must adhere to processes of fair and open competition guided by rigorous peer review. NIJ's research agenda must respond to the real-world needs of victims, communities, and criminal justice professionals. NIJ must encourage and support innovative and rigorous research methods that can provide answers to basic research questions, as well as practical, applied solutions to crime. Partnerships with other agencies and organizations, public and private, are essential to NIJ's success. I work in NIJ's Office of Investigative and Forensic Sciences, whose goal is to improve the quality and practice of forensic science through innovative solutions that support research and development, testing and evaluation, technology, and information exchange. Today's webinar, Emerging Methods for Body Fluid Analysis, is hosted by NIJ's Forensic Technology Center of Excellence, led by RTI International. The center supports the implementation of new forensic technology and best practices by end users and is dedicated to elevating the status of forensic science through advancing technology, sharing knowledge, and addressing challenges. The center also bridges the gap between the scientific and justice communities. Today, you will be hearing presentations on NIJ-funded projects in proteomics, Raman spectroscopy, and micro and messenger RNA, as well as having time at the end for discussion. Our presenters, Dr. Don Siegel from the Office of the Chief Medical Examiner in New York City, Dr. Sarah C. Scholes-Williams from the Department of Forensic Science at Virginia Commonwealth University, Dr. Laurent Ziegler from the Division of Materials Science and Engineering at Boston University, and Tiffany Lane, Master of Science from the Department of Chemistry at the University of Virginia, have all managed excellent projects that we hope can help impact forensic science practitioners in their mission. Thank you for attending this important and informative webinar. Now I will turn the webinar over to Dr. Don Siegel so we can start the presentations.
Okay, my name is Don Siegel. I'm going to give a talk on body fluid identification by proteins. And I would just like to begin by thanking the organizers of NIJ's Forensic Technology Center of Excellence for inviting me to speak in this session of emerging body fluid analysis. I thought I'd begin with a short description of what the New York City Medical Examiner Office is and what we do. We're part of the Department of Health and Mental Hygiene in New York City. The OCME has 5 laboratories, medical examiners where autopsies are done, histology, toxicology, molecular genetics, and of course, forensic biology. The OCME, New York City OCME, is the largest public forensic laboratory in the United States. We test all biological evidence for the New York City Police Department. We have about 10,000 cases per year, 300 homicides, 3,000 sexual assaults, and about 7,000 property crimes. DNA testing is about 50,000 per year, and serology testing used to be about 20,000 tests per year when we were doing presumptive testing. We're also accredited by ANAB and the New York State Commission on Forensic Science. I'd like to begin actually with a description of why serology is still important. I often get this question, and I thought it would be useful to reply to it so that people in the forensic laboratory world are familiar with prosecutors. So from the forensic laboratory perspective, DNA identifies an individual, case closed. But from the prosecutor's perspective, the case is not closed. What they want to know is, what's the source of the DNA? Is it semen, saliva, blood? And especially with increased sensitivity of DNA testing, accused may claim that the DNA profile may have resulted from touch, normal epithelial cell shedding, or secondary transfer. The New York City OCME eliminated serology testing in 2019 because, unlike DNA STR testing, is a presumptive test and does not carry the same scientific weight as a confirmatory test. New York City district attorneys stated that serology testing is vital to their cases, and New York City OCME is now validating our confirmatory proteomic mass spectrometry body fluid assay for casework. Current forensic tests and their limitations, I think everybody probably already knows this. There are chemical tests, enzymatic tests, immunographic tests, and microscopic tests. All of them, except for the identification of sperm under a microscope, are presumptive. I don't think the Kastle-Meyer test is used much anymore, nor is the saliva activity test. One thing that people often ask is, aren't antibodies confirmatory? And they're not. And I'll just give an example why. Antibodies recognize a 3-dimensional molecular structure. This makes their use in the dirty real world of forensics crime scene. I'll repeat that. This makes their use in the dirty real world of forensic crime scenes questionable. So different molecules can have the same conformation, and that's what antibodies recognize. And a classic example of this is in neuroscience, but it's the same as antibodies. On the left side of the screen, you'll see metenkephalin, which is an endogenous pentapeptide. You can see the amino acids, tyrosine, glycine, phenylalanine, and methionine on the left, and you see morphine on the right. Below those two, you can see the 3-dimensional structure of metenkephalin, and morphine. So why does morphine work as an analgesic? Well, they have the same three-dimensional structure. And so when they interact with the receptor, which is similar to what an antibody is, you can see both the metenkephalin and morphine both fit snugly into the receptor pocket. Same thing with antibodies. You can make an antibody to metenkephalin, and if it's directed at the same site as the receptor, it will also recognize morphine. And you can imagine at a crime scene where you have multiple possibilities of bacteria, invertebrates, bubblegum, saliva, everything else that is potentially present at a crime scene, being certain that you've detected what you think you've detected can be questionable. So why use proteins for body fluid identification? You'll hear of other methods being discussed today. The New York City OCM proteins for the following reasons. Back in 2014, Nature published the first draft map of the human proteome. That map has been much more filled out since then. It's been 6 years. And these were 2 articles. It was the cover page of Nature. Basically, what they described was the ability to identify every single organ by its proteome without difficulty of knowing which organ is which. You see that on the left side in A. On the right side of A, you'll see that fetal tissue also has gut, ovaries, testes, liver, heart, etc. Proteomic mass spectrometry can distinguish between fetal tissue and adult tissue. So it's actually an ideal method for identifying body fluids. So the reasons for using proteins are several. Specificity. Proteins have biological functions in each body fluid. Therefore, there are specific proteins in each body fluid that are necessary for those body fluids to function. For example, in blood, you have hemoglobin, saliva amylase, and in semen, seminogelin. We've demonstrated in our laboratory that the assay is sensitive to sub-nanoliter levels. And what I mean by that is levels of the neat body fluid at in sub-nanoliter range are easily detected. We also automatically detect mixtures, something that is not normally done with body fluid testing. If you want to test the body fluid, you need to apply it to one technology and then to another technology and then to another technology if you want to confirm which body fluids are present. Mass spectrometry identifies all body fluids present simultaneously. It's also confirmatory. Proteins like nucleic acids can be sequenced. And when you have the sequence of something, it's confirmatory. Stability. Nucleic acids are far more labile than proteins. Proteins have been found much further back in the geologic history than nucleic acids. There's automatic species identification because, like sequencing DNA, you're sequencing a protein, and the differences, the genetic differences in species are translated into proteins. So you automatically know the species of the sample, and it's amenable to high-throughput analysis. What are the criteria for an ideal protein body fluid assay? So for each body fluid, you should select proteins that are unique to or highly enrich that body fluid, for example, hemoglobin in blood. Sensitivity. Select the most abundant, unique, or highly enriched proteins. And for the main body fluids, blood, saliva, and semen, the most distinguishing proteins are also the ones that are most abundant, and therefore the assay is most sensitive. Also, to improve confidence, we chose to identify 3 different marker proteins in each body fluid. In each body fluid, we also identify multiple sites on each protein. So here, for example, is one marker, seminogelin-1. And instead of just identifying seminogelin-1, we look for peptides in seminogelin-1 that are located in different places. And to be certain that we have identified Seminogelin 1, we must identify all 3 peptides. So for each body fluid, there are 3 proteins, and for each protein, there are 3 peptides. So that's 9 markers per body fluid. On the bottom of the screen, you can see the location of the 3 markers or peptides in green, red, and blue. So this is the peptides that we've chosen for blood, semen, and saliva. As you can see, 3 proteins for each body fluid, 3 peptides for each protein, except for saliva. Both histatin and leg-1 have only 1 peptide, so we identify those. And we find 3 for animals, as well as cystatin. Why mass spectrometry? Mass spectrometry effectively sequences proteins. Mass spectrometry is accurate, sensitive, rapid, and confirmatory. And mass spectrometry is unbiased. You don't tell the mass spec what to look for. There are no specific primers to amplify specific regions. You just tell the mass spec, here's the sample, and the mass spec tells you what's in there. And so for analysis, we don't know or care or presume what the body fluid is. We just put it through the mass spec, and it tells us what's there. Here on the left side of this diagram is our HPLC, which is an exigent nanoflow. And on the right side is the mass spec, and the red line connects the column that separates proteins before they enter the mass spec on the right. So the exigent HPLC works by separating peptides. The peptides enter the column on the left at the top of the screen. They get separated. As you can see, the red and blue are together at the beginning. They get separated over a C18 column, and after they separate, they go into the mass spec. And so on the right, you'll see hemoglobin peptide, SVA, is separating from other peptides. And you can see on the bottom the signal and the retention times of each peptide. Once it enters the mass spec, over here you'll see the whole bunch of peptides coming into the mass spec simultaneously. The window is opened just for the mass of the peptide we're looking for. Once it's in there, it's fragmented. And each fragment of this same exact hemoglobin peptide, 4 of those fragments are identified. Their masses are known. And once all 4 of those transition ions are identified, we know the sequence, and we can say that this peptide came from hemoglobin, and we know its sequence. And so there are 3 proteins per body fluid, 3 peptides per protein, and 4 transitions per peptide. This is the workflow. It's relatively simple, at least on paper. Actually, it's not very difficult. Starting at the left, number 1, samples, tissues or swabs, whatever is, fabric, whatever comes into the OCME is extracted. The protein is extracted. It takes about 30 minutes. After extraction, we do a quant. The quant takes about an hour. After quanting, we digest it. And currently we digest overnight, but we're working on a 3-hour digestion assay that looks pretty promising, but it will not be in the initial validation protocol that we're working on now. After digestion, the peptides are introduced to the HPLC. They're separated. They go into the mass spec, which is not a MALDI anymore. We're using now a Q-trap. After mass spectrometry, the peptides are sequenced, and after sequencing, we search them in a database. So once the sample is introduced into the HPLC, it takes about 20 minutes from beginning to end to identifying the peptide. You can do 1 sample at a time if you have an emergency, or you can do a 96-well plate. And our current assay is designed to do about 400 samples per week. Currently, the assay that we're working on is under validation. Once we complete the validation, which we hope to do sometime this year, we'll present it to the New York State Commission on Forensic Science for approval. One thing I should also note is that we've also found markers for menstrual blood and vaginal fluid, which are occasionally of interest in forensic situations. And those proteins will come online after the three major body fluid proteins. I'd like to thank a lot of people who have been working on this for quite a long time. At New York City OCME, Dr. Hayi Gang, Tatiana Perez, Iman Al-Mubarak, and Jahaira Zapata, and Samantha Munyor, Erin Butler, and I would also like to thank David Fenyo. And of course, this work could not have been done without the generous support of NIJ. Thank you.
Well, it's my pleasure today to be part of this webinar sponsored by the Forensic Technology Center of Excellence, allowing us to talk about our latest developments in using surface-enhanced Raman spectroscopy to develop a platform for rapid identification of human body fluids. Of course, the motivation for such a platform might be obvious to everyone, but let me just talk about what I see as the value of such a SIRS-based platform. And that's largely to develop a single platform that can detect and identify body fluids. And body fluids we're interested in are blood, semen, vaginal fluid, saliva primarily. And these, of course, are important common evidence at crime scenes. And the idea is to provide rapid here means on the order of minutes, confirmatory identification of body fluids and their mixtures. We'd like the technique to be portable for use, of course, at the crime scene, but also in forensic laboratories. You want the technique to be sensitive to be able to identify trace amounts of body fluids. You'd like it to be non-destructive for downstream DNA testing, for example. You'd like it to be relatively inexpensive, easy to use for non-specialists. And of course, the point is that I think that these goals could be achieved by a SIRS-based platform. Just so that everyone is in the same place for this brief discussion, let me identify Raman spectroscopy as the inelastic scattering of light, which you see illustrated here. The instrumentation for Raman spectroscopy is pretty straightforward. One has a laser excitation source, as I'm showing here, and then the scattered light is collected and the frequency components are analyzed by a spectrometer. Just to give some example, this is what I'll call the normal Raman spectrum, that it's not a SIRS spectrum. It's the normal Raman spectrum of dried whole blood when excited by 785 nanometer excitation. So why use Raman? Well, it has a number of advantages, I believe. The unique molecular signatures for ID can be identified by Raman spectroscopy. The inherent vibrational motions are the labels themselves, so to speak. There's no need for extrinsic labeling or reagents. The vibrational features are narrow, and that's highly valuable because now you can have multiple unique markers for a given molecule, which of course increases the reliability of the technique. And then it also allows the technique to be a multiplexing technique. That's to say that you can get the unique molecular signatures from a number of molecules that are in the illuminated region. And that, of course, is ideal for complex biological mixture analysis. The sample size is small. It just depends on the illuminated laser-focused laser region. It's easy to use, it's rapid, it's non-destructive. However, there are some limitations. Raman is a relatively weak effect because it's a two-photon effect, light in and light out. And so that generally limits one to looking at samples that are in high concentrations. And it also means that you have to use higher laser intensity. Another limitation potentially is the overlapping of inherent fluorescence, which can be quite bright and broad and overlap the signature that you're looking for. And then another feature in normal Raman spectroscopy, which can be problematic, especially for dried body fluids, is the spatial heterogeneity that you can find in such dried body fluid stains. And so, it may be that the signature that you get in different regions can be quite different. So I'm going to describe for you the way that surface-enhanced Raman scattering can overcome these limitations. And first, let me just very, very briefly describe what SIRS is. And it's that molecules that are close to nanostructured metal surfaces can show Raman spectra that are enhanced by 10 to the 6th or 10 to the 8th. So these are enormous enhancements, not factors of 6 or 8, but 6 to 8 orders in magnitude. The key is that the excitation has to be chosen to use, which is the right color, that has to be resonant with the surface plasmon resonances of the nanoparticle structure. And so the way to think about this, just very, very briefly, is that the nanostructured metal surface acts like a local electric field amplifier. And the electric field, of course, we're talking about is the radiation. So this effect arises due to a resonance condition between the incident light and the scattered light and the surface plasmon excitations of the nanostructured metal surface. In addition to this, this main surface plasmon resonant source of Raman intensity, there also can be chemical enhancement effects, and these arise due to new charge transfer excitations that are created by molecules being close to nanostructured metal surfaces. And this can contribute a factor of 10 to the 2 or 10 to the 3, smaller effect in the overall enhancement, but it can be very important in controlling the pattern of relative intensities, as you'll see. So why use SIRS for forensics identification and detection? Well, it has all the advantages of Raman spectroscopy that I described, plus it provides now high sensitivity, which will allow you to look at much lower concentrations down to particle per million, as you'll see. Fluorescence is quenched. It's not a problem anymore due to rapid energy transfer to the metal surface. And the fact that you've got this enhanced sensitivity allows you to look at solutions of the body fluids. And so that can really reduce the problems with heterogeneous spatial samples. Here's a picture of the SEM of the surface of the SIRS substrate that I'll be talking about exclusively today. We make them in our laboratory. They're in situ grown, nanoparticle covered little bits of glass, SiO2, produced by metal ions doped sol gel procedure. We can make them with gold or with silver. And basically you have a piece of glass, and it's just a millimeter or so, that is covered with clusters of these gold or silver nanoparticles, as you see here. The technique in itself is really very straightforward. We drop a microliter of some solution, whether it's saline or water or something else, as I'll describe to you in a couple of minutes, onto the suspected body fluid stain. We then suck that volume back up into the pipette, put it in on the SIRS substrate, and in fact, we've built little chip holders that match the footprint of a handheld Raman instrument, which you see pictured here. And then, of course, is then just connected to a computer for classification analysis. So it's a very straightforward and easy technique to use. So here are some of our earlier work for SIRS spectra of dried human body fluids. These are dried for 24 hours, and all of these are 30 spectra from 2 donors. And the point that I want to make here is that you have pretty good signal to noise. These shaded areas give you plus or minus 1 standard deviation. There's no fluorescence. And there are clear differences between the SIRS spectra of these different body fluids. The classification of these different body fluids will be carried out for everything that we're going to talk about today anyway, a technique that employs what's called partial least squares discriminant analysis. And this is very standard, and the only trick, so to speak, on this is that we convert the SIRS spectrum to a barcode for input for the analysis by this classification procedure, where we just take the sign of the second derivative as our input vector. And what I'm demonstrating here is the output of this PLSDA classification procedure can be quite successful, even in our preliminary work. identifying these different body fluids. And identification then would be determined by group membership in a series of library spectra. And so in this training set, we've got 98% sensitivity, that's analytical sensitivity, and 99.45 specificity. Okay, but there's something here I didn't like. And that is illustrated here for the Search spectrum of dry blood. I mean, what is the compound that distinguishes blood from any other body fluid? And the obvious answer is hemoglobin. And when we were using saline or water, these are the components that dominated the Search spectrum. And so I wanted to find a procedure that would bring out the hemoglobin content of blood. And so we were inspired by the classic Teichmann test from 170 years ago, which detected crystals of hemin, and they used glacial acetic acid to tease the hemoglobin moiety out of the protein structure to allow the formation of these crystals. And so what we found is that if we use 1 microliter of 50% acetic acid on the suspected dried blood stain, we got fabulous results. And let me demonstrate that for you here. By using this 1 microliter of 50% acetic acid, our signal intensities were increased by at least an order of magnitude. And the reproducibility was also tremendously enhanced. So this was a big step forward. Here are Search spectra for a number of these body fluids, including blood. And here are the classification results. They're perfect, 100% sensitivity, 100% specificity analytically. And then when we challenged this training set with a series of unknowns, these are 20 unknown spectra, they all identified properly via our PLSDA barcode procedure as human blood correctly. Sensitivity, so what is giving rise to the signature? Well, again, I described our procedure as the optical analog of the Teichmann test. The molecule that we're looking at is something like hematin, that's to say it's a hemoglobin degradation product. It's a MET ferric iron center. The heme portion has moved out of the protein pocket. And most interesting, what's most interesting to me was that the Search spectrum didn't resemble at all the normal Raman spectrum of hemoglobin that's illustrated here. It looked a lot like hematin, which is the molecule that you see pictured here. And all of these bands could be properly identified, but I don't have time to go into that. One very fascinating aspect about this is that the Search spectrum of human blood on gold was almost completely different than the Search spectrum on silver. And that's illustrated here in the difference between the red and blue spectra. Here, I've highlighted that difference by blowing up the region between 1400 and 1700 and normalizing, just showing you how different these two spectra are gold and silver. And that's something that we have completely understood now. That's due to the differences in the metal to pi star charge transfer character on gold versus silver. And so what I'm demonstrating here is a very powerful consequence of the chemical enhancement effect. What about sensitivity for detection and identification? Well, one way to demonstrate that is we made diluted dry blood samples of whole blood, dried for 24 hours. We used our acetic acid procedure with 1 microliter. And as you see here, we can get a fabulous Search spectra down to dilution of 10 to the 5. Clearly, these are regions that are not evident to your eye from a dried blood stain. No normal Raman spectrum, of course, can provide any signature at these dilution factors. We're sensitive to less than 10 picoliters of total blood volume, or about 10 nanograms. The sensitivity is greater than that of RSID techniques, and it's comparable to immunochromatographic tests. Here's an example of Search sensitivity That also introduces another technique for use in forensic science. Here's a 1 square foot piece of glass before we've done anything to it. In this second glass that I'm showing you here is the same piece of glass, and we've smeared a milliliter of water that had 10 microliters of blood on it over this glass. And of course, the point is that you can't see any color. We've dipped a cotton swab in our 50% acetic acid solution, swiped a 1 inch by 1 inch area of glass, touched that swab to a SIRS chip, and boom, the spectrum just pops up. So there's tremendous sensitivity, and that's just one anecdotal example with a swab technique. Here we've placed dry blood on different surfaces. You can see the range of surfaces that we've dried the blood on for, this is up to a week. They're porous and non-porous materials. We've used our swab technique, and you can see you get gorgeous and consistent SIRS spectra of blood showing up from this procedure. Here's another example showing how we can discriminate blood from 31 reddish brown stains. We've acquired SIRS spectra from all the materials and substances that you see here, set up a classification procedure, and then challenged it with dried human bloodstain SIRS spectra, and all 25 in our challenge group, of course, properly identify with human blood. Here's another application, making sure that we can identify the blood stain as human as opposed to animal. We proceeded initially with our standard 50% acetic acid procedure. And, you know, it didn't work very well. We got misclassifications. And when we challenged that classification procedure with an unknown, it did not work. And it's, of course, not terribly surprising because it's it's hemoglobin that is giving rise to this signature. So we adopted the procedure, and instead of using 50% acetic acid 1 microliter, we use a 10th molar ammonium chloride in ethanol, a different pH, and that, of course, is and different solvent character, and that's going to solubilize different components in the dried blood stain. And using that procedure, then it works, SIRS works perfectly. Here we're asking the question, is the unknown blood stain human blood or animal blood? And of course, all of the unknowns classified properly as human. And in fact, we can even use our classification procedure to identify species, and challenging that again with the human blood, and it captures the correct classification. I think the final application I'm just going to talk about, of course, if you have a technique which is so sensitive that your eye can't find it, how can you employ it effectively to make a blood identification, for example? And of course, as probably all of you know, the standard presumptive ID test in a crime scene is to locate blood with the blue glow of luminol. This is a complicated chemical process, and I'll just give you one example that demonstrates how we can effectively use this. Here, what we did, this is a 1 foot by 1 foot ceramic tile that we've put 1 milliliter of blood on, let it dry, came back and cleaned it up so there was no trace of blood that your eye could possibly see. And then we, of course, exposed that to luminol, and it lit up blue. So here's the corresponding SIRS spectra for these different situations. I'm showing you here in blue the luminol SIRS spectrum, and unfortunately it gives a decent SIRS spectrum. Of course, speaks to the sensitivity of SIRS for various molecules. The red spectrum that you see here was just the SIRS spectrum obtained by a swab of the tile after the cleanup. And again, it makes the point that we're sensitive to make a confirmatory identification of dried blood, even when your eye can't see it. And this spectrum at the top is the surface spectrum that we obtained after the blue glow of luminol identified that tile as having blood on it. And here, you can certainly see that there are features that are coming through that are coincident with the surface spectrum of blood. When we challenged those spectra that were obtained from the ceramic tile that was already exposed to luminol and threw it against these different body fluids, those spectra correctly identified with blood. So even though there was no evidence of blood on the tile by eye and was located by the blue luminol gold, certainly our surface spectrum confirmed that there was blood in that tile, and this was not a false positive as one might get from just bleach being on the tile. So in conclusion, I think that we've demonstrated that 785 nanometer SIRS can be a sensitive and rapid portable platform for confirmatory identification of trace amounts of human body fluids. We can alter our protocols to bring out different components of the complex biological fluid, as demonstrated, for example, in particular for the animal blood. We've identified what the molecular origin of these signals are. Something I haven't talked about but was implicit in what I was showing in here is that donor variability is much smaller than the difference between the characteristic surface spectra that identify different body fluids. And in terms of future work, I think the most important component of our future work is to get this technology into the hands of forensic investigators to get feedback from practitioners in the field and in the laboratory. So I must conclude by thanking the folks that did the work. You see folks who carried out these studies and have taught me quite a lot about forensic science. And I also need to thank in particular the National Institute of Justice for funding this work. Thank you.
Today, I will be presenting data on two projects that use messenger RNA as the targets that are being performed in a lab at the University of Virginia. So why use RNA? Well, one, the DNA material is being used for DNA profiling, so we don't want to take up any of that. Two, there are many different types of RNA in the human body that we can take advantage of. For these projects, we're looking at messenger RNA, which carry the code for protein translation and are uniquely secreted based on cell type or region of the body. There are many advantages to using messenger RNA. Research has shown that messenger RNA are stable in fresh and compromised samples. There are many commercial kits that provide a one-step coevolution of DNA and RNA, saving time and cost for laboratories, as well as emerging technology allows research of messenger RNA on various platforms. These platforms have shown better sensitivity compared to conventional methods, as well as better specificity of the messenger RNA targets. One of the big advantages of these platforms, of this technology, is that multi-fluid detection can be obtained in one assay versus multiple assays. Messenger RNA research is primarily used for body fluid identification in the forensic realm, but there is publications on sex determination or age of stains and wounds. So the first project I'll talk about is using an isothermal amplification method. We have published on a Y-screening DNA target. We've also looked at messenger RNA body fluid targets. We've compared and contrasted colorimetric and fluorescent indicators in the LAMP assay, and we are developing an integrated one-step heating and analysis device. LAMP differs from PCR in a few different ways. First, it's isothermal. Second, it uses 4 to 6 primers instead of the 2 primers of PCR, and it produces multiple size amplicons. So after the first 4 primers anneal and extend, a key-like structure is made. And then from here, the other 2 primers allow for these multiple size amplicons to be produced. LAMP can be used with colorimetric or fluorescent dyes, and results are obtained in less than 60 minutes. For this project, we combine LAMP with an image analysis method. This allows for simple quantitative detection of the color change, allowing for a threshold to determine if a reaction is positive or negative. I will show you data on messenger RNA targets that differentiate venous blood from menstrual blood, as well as semen, saliva, vaginal fluid, and some of the data from the Y screening DNA assay. So we wanted to create universal conditions using LAMP. First, we looked at the lysis and purification method. We used the RNA-Z mini kit, and we added on the on-column DNA treatment. When we did that, we obtained specific amplification. Next, we optimized the LAMP assay. We first looked at the various temperatures. We settled on 63 degrees that allowed for amplification of all of the targets. We then looked at primer concentration, where we noticed that the body fluid target, the blood target, was more sensitive than the rest of them. And so we decreased that primer concentration, but we were able to increase the rest of the primer concentration body fluid targets, which allowed for amplification in 30 minutes. From here, we looked at specificity against other fluids. We were able to distinguish between all 5 of the body fluids. We also were able to distinguish the Y screening DNA target in various samples. Lastly, we looked at sensitivity. We were able to show that total RNA in the picogram range consistently amplifies all 5 messenger RNA body fluid targets. We're also able to show that the Y screening DNA target can amplify in 50 minutes, 66 picograms of DNA. From here, we wanted to further validate our LAMP assay using a mock study. We had various samples containing cross-reacting fluids like breast milk and nasal mucosa. We also had various mixtures on different substrates, postcoital swabs, as well as age samples. When we look at this data, we were highly encouraged by the saliva and vaginal fluid results. However, we did need to troubleshoot the other 3 targets to determine why we had less than wanted results. When we did this, we found that the venous blood target is non-specifically amplifying. We found that the semen target could not amplify lower concentrated samples. And when compared to PSA, the PSA card did do slightly better. The menstrual blood target we found was amplifying vaginal fluid samples, and so that's another target we're looking into. Overall, we can show that the saliva and the vaginal fluid markers are working very well in the LAMP assay, and with a few more tweaks, we will be able to get the other 3 targets up to par. We've also developed an integrated device that allows for isothermal heating and automated image analysis. This device has a 96-well plate format, so a scientist can use a plate or individual reaction tubes. The data comes out in real time, and so by a graph, you can see the change in hue, and then once the tube location lights up green, that tells the scientist that the reaction has crossed the threshold and is a positive reaction. We have data on this device showing that within 10 minutes, we can get to the necessary temperature and hold for the required amount of time. We have also compared the temperature in this iLAMP device to a conventional thermal cycler, and we've seen consistent results. Next, we did testing of the mRNA markers in this device, where we show that we can sufficiently amplify samples and not amplify negative controls. The stable hue at time 0 allows for thresholding and determination of a positive versus negative reaction based on hue. When compared to the thermal cycler, we did see consistent results across all of the samples. We further analyzed some of the samples from the mock study. While the results are not what we expected, we do believe that we can further optimize this device to get it up to par. So here we are showing a device that can do 1-step body fluid profiling in 60 minutes. It has consistent heating and image analysis of all of 96 reactions. So in summary, we have developed a universal method for 5 different messenger RNA targets and a device that can do it in 1 step. Future efforts are to look at other messenger RNA targets and to further allow multi-fluid detection of 15 samples simultaneously. We've also expanded the LAMP assay to include clinical applications, as well as adaptation to microfluidic technology. Next, I will present data on a project that integrates a published body fluid panel with microfluidic technology. You'll see data on size separation, RT-PCR, and future efforts of coevolution of DNA and RNA. The Institute of Environmental Science and Research in New Zealand have developed 3 different panels that allows for body fluid profiling. The quadruplex panel differentiates the bloods. The pentaplex panel contains the sexual assault messenger RNA targets. ESR's protocols are similar to current forensic laboratories, and so there's no new chemistry here, but they have shown that these mRNA targets are not only fluid specific, but human specific as well. They further validated these panels, showing that with less than 1 microliter of liquid body fluid, they can detect all 11 targets, as well as they've validated using various mixtures, postcoital samples, and age samples up to a year plus. For our project, we are combining these panels with microfluidic technology. Here at the University of Virginia, we have expertise in this area, developing systems that will rotationally drive liquid movement inside chips. These systems can do anything from lysing cells all the way to fluorescent size separation of various fragments or colorimetric detection of dyes. We've used these systems for various forensic applications, most notably human DNA profiling. We've shown that we can perform a 10 loci PCR in less than 15 minutes. And we can also fluorescently size separate those 10 loci in less than 5 minutes. So this project is combining these 2 techniques. First, we looked at on-chip separation. In most forensic labs, DNA profiling is performed using a 3130 or 3500 instrument. The idea here is to take the functionality of this instrument, put it onto a micro device with the same or better sensitivity of fluorescent detection. We designed and fabricated 5-layer chips that allow for 3 samples to be separated per chip. All the scientist has to do is input a sample and input polymer to the chip. After a few rotations, you're ready for microchip electrophoresis. We did need to optimize a few of the parameters. First, we looked at injection time to allow for the highest RFU in the 3 fluorescent channels. We also looked at if heating was needed of the microfluidic channel. We also looked at if heating was needed in the microfluidic channel, which it is not. From here, we started separating some of the single fluids received from ESR. What you're looking at is the comparison data between on-chip separations 3130 and 3500. What we found was that in the duplex, there was peak imbalance looking at the 3130 and the chip data. We found some peak imbalance in the chip data using the quadruplex panel. And as far for the pentaplex panel, the KLK2 marker was the lowest marker detected across all 3 platforms. However, we were able to show consistent size separation using the on-chip separation, and it was similar to both the 3130 and the 3500 results. We also separated a few mixture samples. The first one contained vaginal fluid and semen. So this size separated all 5 targets in the pentaplex panel, which we were able to consistently do on chip. The second mixture sample contained vaginal fluid and buccal. This size separated 3 different targets in the pentaplex and the duplex panel. And again, we were able to consistently amplify this target, consistently detect this target using the on-chip platform. So overall, we found that by adapting size separation to on-chip, we are not only getting consistent results on-chip, but it's similar to the gold standard 3130 and 3500 instruments. From here, we started to optimize the RT-PCR protocol published by ESR. While there are many different commercial kits, they use a 2-step protocol that uses a hot start tack. This was performed in a conventional thermal cycler that most forensic laboratories use. Most thermal cyclers have ramp rates of approximately 3 to 5 degrees per second. The idea here is to use this same functionality and put it onto a microchip as previously described. This is an image of our system where the red arrows are pointing to Peltier heaters. Here we're using a dual system, so our system will clamp down and sandwich the disc, allowing for consistent heating of your reactions inside the chip. One of the major advantages of using a dual Peltier heaters is that the ramp rate is approximately 8 to 9 degrees per second, which is double what the thermal cycler can do. And so it allows us to achieve RT and PCR on chip. We designed a 5-layer chip that allows for 6 RT-PCR reactions to occur per chip. All the scientist has to do is input the RT reaction into the RT chamber, input the PCR reagents into the PCR chamber. Once the RT reaction is heated, it will flow down an aliquot into the PCR chamber. The scientist can pull off excess cDNA to do interim analysis if they prefer. The PCR reagents are then flowed into the PCR chamber, and heating will occur to create your target amplicons. So we looked at the RT protocol that was published by ESR, which is what you're seeing on the screen. We went through this stepwise to decrease the time for each step. This first step, we were able to decrease from 10 minutes to 1 minute without decreasing the cDNA production. What you're looking at is the CT values of in-tube reactions versus on-chip reactions. Next, we looked at the 37-degree step, which is currently 2 hours. We were able to get that down to 8 minutes with minimal change in the cDNA production. Lastly, we looked at the 85-degree step. This we were not able to decrease, and we did not see any cDNA production. So this stayed at the 1 minute. Overall, we were able to take the RT protocol from 2 hours down to 10 minutes by adapting it for on-chip. Lastly, we looked at the RT enzyme, which is currently a 1x in tube. When we increased that, we really did not see a change in cDNA production, so we stayed with the 1x concentration. Now that this protocol was optimized, we started analyzing RNA lysates received from the ESR lab. The solid bars indicate results from in-tube. The striped bars indicate results from on-chip. What we concluded was that all of the fluids were able to be detected before a CT of 30. After a CT of 30 is when we saw the negative controls from this assay. By adapting the RT protocol onto on-chip, we only take a hit of 3 to 5 values in CT. So we were very encouraged by these results and continued the optimization. Currently, we're looking at the PCR protocol. This first step is for enzyme activation. In tube, it's 15 minutes. We're able to get that down to 4 minutes with sufficient amplification of the messenger RNA targets. Our ongoing optimization is decreasing the time for each of the remaining steps with a goal of less than 45 minutes for PCR. I have no doubt that we will achieve this. Previously, we have shown that we can do rapid PCR. This is 30 cycles within 13 minutes. So the data from this project shows that we can separate 3 chips per sample 4 times faster than the conventional methods. When we apply RT to on-chip, it's 13 times faster, and our current optimization of PCR will have it in less than 45 minutes or 2 to 3 times faster than in-tube. So in summary, we have a project using a LAMP assay that allows for automated real-time results in less than 45 minutes for 5 messenger RNA targets. We have a second project that allows for RT-PCR and fluorescent size separation of body fluid panels on chip that is 4 times or 13 times faster than conventional methods. Overall, we're really excited about these projects and the applications for the forensic world. I'd like to thank James Landers and current and former grad students for all of their help on these projects, as well as the scientists at ESR and New England Biolabs and FTCOE for allowing us to present this research. Thank you.
Hi there, my name is Dr. Sarah Williams. I'm an associate professor at the Department of Forensic Science at Virginia Commonwealth University. And my research is focused on developing new and better methods for body fluid identification, as well as the front end of the forensic workflow. And so microRNA analysis is a pretty big part of my lab. It's one of about 3 major projects that we have going. And so today I'm going to talk about exploring the utility of microRNAs as potential biomarkers for body fluids. So just as with all of the other projects that are being talked about today, this project has been supported by a couple of different grants through the National Institute of Justice, and my opinions are mine alone and not reflective of the Department of Justice's opinions. So let's start by talking a little bit about what microRNAs are and what they do, because you may not be that familiar with them. So microRNAs are small, non-coding RNAs. When they're mature, they're about 18 to 22 nucleotides long. They're single-stranded, and they're encoded with, or they're encapsulated within a protein complex called the RISC complex that's out in the cytosol. And the purpose or job of the microRNA is to kind of fine-tune or attenuate protein translation. So what happens is at the point of translation, where the ribosomes are translating that messenger RNA into protein, the RISC complex with its microRNA single strand find a complementary sequence or partial complementary sequence. And usually that sequence is either on the 3' end for the most part, but sometimes on the 5' end as well. And what happens is that if it's a perfect match, then the messenger RNA is cleaved and degraded and no longer able to be translated. And if it's an imperfect match, the RISC complex just kind of sits there and blocks translation sterically, just by blocking the translation from finishing. And so either way, what we end up getting is a fine-tuning of or attenuation of protein production within the cell. Now, if you think about how we know that there are a lot of different types of tissues in our body, and so we're going to have microRNAs that are tissue specific because, of course, different proteins are needed for different tissues. But we're also going to have ubiquitous microRNAs, which work to fine-tune that protein production for normal cell processes that all cells need. And so ideally, for body fluid identification, we have microRNAs that are tissue specific, and we also have microRNAs that are pretty ubiquitously expressed. Now, another advantage of potentially using microRNAs for body fluid identification is that they're very stable. So between the small size and the fact that they're always encapsulated within a protein or packaged in a lipid vesicle, that makes them very resistant to degradation, as I'll show you in a little bit. And then finally, another really distinct advantage for microRNAs as a potential body fluid identification method is that they're detectable in DNA extracts. So they co-extract with most of the DNA extraction methods that we use in forensic biology. And so the possibility of detecting them without having to do an additional extraction method is quite promising. Now, disadvantages I'll talk about as well. So analysis methodologies are challenging for something that's that small. 19 to 22 base pairs isn't a lot of real estate for us to work with. And they also tend to be conserved across species. So it's going to be difficult finding a microRNA that is species specific. However, expression seems to be very different from species to species. Now, I'll talk just for a second about the methods that we use to analyze microRNAs. The majority of the methods that you'll see in the forensic literature are quantitative reverse transcription, qPCR methods. And again, there's some complexity in building the primers. You can't fit 2 primers on a 19 to 22 nucleotide target. And so you'll find that most of the primer strategies are actually extending the microRNA to a longer species, a longer sequence, so that we can amplify that more easily. Now, with high-throughput sequencing, that's used for discovery of new microRNAs, discovery within clinical studies, but not used as much in forensic analysis, although a couple of studies, including one of ours, has used it. And then microRNA analysis you'll find almost exclusively within the biomedical research area. That's a very popular analysis method for biomedical work, but since we don't have that type of methodology in the forensic laboratory, it's not likely to really catch on in forensic work. So this is the Q-script by QIAGEN. This is their approach. You'll find that the TACMAN approach is very similar. So they're extending the length of the microRNA with an additional primer, and then with that longer sequence, they're adding in their forward and reverse primers and their TACMAN probes. Now, in terms of the forensic literature, microRNA analysis has been somewhat inconsistent from researcher to researcher, and it's not because there are any errors, really. It's more because the platforms are very different. So the analysis methods from manufacturer to manufacturer can give some differing results. But the bigger reasons are normalization approach and sample sizes. So what we choose to normalize or control, but our body fluid specific microRNAs with is going to dramatically impact the relative expression. So whenever you've got an RNA, whether it's a microRNA or a messenger RNA, you usually want some type of endogenous, ubiquitously expressed control RNA that you're also measuring to make sure, number one, that you're getting results if you should, but also to normalize that expression. So some researchers have used U6B, which is a small RNA. It's not a microRNA. Other researchers have used miR-144 or LET7G or LET7I or combinations of multiple microRNAs. And so that's going to dramatically impact the relative expression values of the body fluid specific microRNAs. Now, another potential reason that we see differences from study to study is the sample sizes. So many of these initial studies are microRNA discovery studies. And so the sample sizes are relatively small, let's say 5 to 10 samples of each individual body fluid. So maybe 5 to 10 samples of menstrual secretions, 5 to 10 of blood, 5 to 10 of semen. Now, certainly that adds up and can provide significant preliminary data. But when you start analyzing a larger population, say 50 or 100 individuals, sometimes those expression, they don't follow through. We don't see the same data with a larger population sampling. So for discovery of microRNAs that may be body fluid specific, most laboratories, including ours, go through a similar workflow to this. So when we were looking at identifying microRNAs that were characteristic of different body fluids, we pooled multiple body fluid samples from different donors and did parallel high-throughput sequencing of the small RNAs within those body fluids. And ultimately, we identified all of the detectable microRNAs in each sample. And that included both microRNAs that were diagnostic for each body fluid, but also potential microRNAs that were ubiquitously expressed. And so we did that through high-throughput sequencing. And then we followed through with verifying those results with qPCR analysis. Now, the problem there is that when you switch platforms, you're not going to have perfect corroboration. And some of that is because qPCR analysis is significantly more sensitive than high-throughput sequencing in most cases, because again, you can dial down to 1 or 2 copies of any of these microRNAs and amplify them up using qPCR, but you may miss them due to sequencing depth when you're doing high-throughput sequencing. And once we actually identified about 8 microRNAs, 6 of those that were characteristic for different body fluids, and then 2, LET7G and LET7I, that we found to be excellent endogenous controls. We actually averaged the data together and then for those endogenous controls. And we did a large population study set. So we started off with smaller sample numbers of each body fluid, let's say 20 samples of each body fluid. And then we moved up to 50 to 100 samples of each body fluid. And we did a tiered approach and evaluated the data at each step to conserve reagents. Because when you're testing, let's say 50 samples of each body fluid against 8 different microRNAs in duplicate, that starts to add up really quickly. And so ultimately, we identified 4 microRNAs, miR200B, miR10B, 26B, and 891A, that could help us to classify almost all of the body fluids. So blood, semen, vaginal secretions, menstrual secretions, saliva, feces, and urine, with a pretty high accuracy. Once we identified those, we then tested the data for reproducibility and accuracy. We tested for expression over biological time, which I don't have time to show here, but that's, for example, if miR200B is our diagnostic microRNA for blood, do we see expression, a similar expression in the same donor if we take blood from them 3 times over the course of the week? Or more importantly, the 21 days that a woman is not menstruating, do we see the same results in vaginal secretions? Do we see the similar expression levels across the 4 to 7 days that a woman is menstruating? And so that was really important to us. We looked at organ and tissue analysis and species specificity. And then finally, we looked at stability. And I don't have time to talk about all of those, but I will be talking about some of those. So overall, we found that accuracy using a quantitative discriminant analysis was overall 92.5% accurate. And so you can see that if you look down, so the columns are the actual body fluid identities, right? And so blood on the left, we've got 45 samples that were blood and characterized as blood. But then when you move down, 4 samples that were actually blood were incorrectly classified as menstrual blood. And so I've highlighted the samples that were incorrectly classified for each individual body fluid. What we found was that for the most part, blood, feces, menstrual secretions, saliva, and urine were very highly classified accurately. We did see some issues with semen and vaginal secretions that we're still working on, and trying to identify other microRNAs that might help us to flesh that out a little bit better, because we know that those are 2 of the most important body fluids for us to accurately classify. But we were quite impressed by our ability to pretty accurately classify those. Now I want to talk a little bit about the stability of microRNAs. So microRNAs have shown themselves to be highly stable under compromising treatments. So this is blood, saliva, semen, and urine. In the top inset, it's been treated at 95 degrees and 55 degrees Celsius over time, and you see very little difference in expression. And then if you look down in the lower inset, we're treating blood, urine, semen, and saliva with ultraviolet light, a couple of different concentrations of bleach, dish detergent, acetic acid, and some heat. And you can see that we do lose some of the samples, urine and saliva specifically, when we treat with high amounts of bleach. And some loss with dish detergent, but otherwise the species are highly stable. Another thing that, again, I feel is really important and is the objective of our current NIJ grant is taking the results that we found with RNA extracts and applying that to DNA extracts. So we know that microRNAs co-extract with DNA extracts. There's a couple of papers that I've put in the resources where you can see that we tested traditional organic, DNA Investigator, DNA IQ, and found that we could accurately predict body fluid specific microRNAs using DNA extracts, not RNA extracts. Now that's primarily because they are, again, encapsulated, whether it is in a protein or if they're exported from the cell in exosomes, which are lipid vesicles. But either way, we we corroborated other researchers' findings that these microRNAs are co-extracting, usually with basic silica binding extraction methods, but also under organic extraction methods. Now, for potential casework use, I just wanted to give a quick example, although most of you can come up with plenty of examples. So if we have a clear stain on a shirt, we could certainly do serology, but it's going to only indicate the origin of the stain that you're testing for. So if you're only testing for semen and you're not testing for saliva or vaginal secretions, then you're not going to see that. And so you could take a small portion of your DNA extract and use it for microRNA body fluid identification via RT-PCR. And that would, that data would go automatically into an Excel spreadsheet that's going to give you a prediction of what body fluid is present and what the probability is of those body fluids. And hopefully we're working on hopefully being able to indicate if a mixture is present. Then you take that same DNA extract, of course, and develop the DNA profile. And so you have developed a DNA profile and identified the body fluid present with a % confidence in that body fluid identification. Now, in terms of estimated false positive and false negative rates, our classification right now is at a 92.5%. And so we expect 92 to 95% true positive calls, more false negatives than false positives. And so far we've tested intimate samples and been able to get data at least to about 5 days. Now, a disadvantage is that we're still working on identifying if we can differentiate what is going on with mixture samples. And so that data is definitely forthcoming, but I don't have it for you right now. I anticipate it may be an issue, but it may not. So in conclusion, while microRNA analysis is certainly not ready for prime time, ready for casework yet, we're getting very close. We've demonstrated, multiple research groups have demonstrated tissue-specific and stable expression over different ages, ethnicities, and across time within a single person. They're highly robust, they're very stable, and they're detectable in DNA extracts, which gives them great promise for easy integration into the forensic science workflow in the future. There are certainly multiple detection platforms that we can use for this. We could even move into adding them as part of a panel in high throughput sequencing. But there certainly is future work to do. So the community needs to work towards standardization. So selecting microRNAs that work on a given platform or multiple platforms, select a normalization strategy. So are we going to normalize? Which microRNAs are we going to use to normalize? Certain body fluids do still give us some issues. So for us, it's semen and vaginal secretions. For other researchers, I know vaginal and menstrual sometimes give issues. And many other researchers haven't looked at urine and feces. And so we need more researchers to look at the other body fluids as well. Again, as I mentioned, mixture interpretation may be an issue, but we're still working on that. And then moving forward are the challenges of potentially multiplexing microRNA. And again, with such a challenge of developing your primers, that could potentially be complicated. And so we may want to think about a high throughput sequencing approach, but we'll see where the field goes. That's all I have. Thank you for listening. I want to thank RTI and the Center of Excellence, the Department of Justice, and VCU's Department of Forensic Science, as well as probably 25 different students that have worked on this project over the course of 8 years, especially Kara Lewis and Sierra Rhodes, my 2 PhD students. Thank you.
All right, let's dive into some questions from the audience. First one that I have here is for Dr. Siegel, and it is a pretty straightforward one. How do you separate the proteins from the DNA?
So there are multiple ways to do it. In the previous grant that you can use any of the QIAGEN available kits, and which separate RNA, proteins, and DNA. Currently, we're just using the source itself. So if it's a matrix of fabric or whatever, we get a cut of it. But we did look into using simultaneous extraction. The problem with that is those extractions, when you do multiple nucleic acids and proteins, the problem with that is the method that's designed solely for DNA still gives the best signal for STR profiling. So there was a loss of signal in STR profiling. Could that be worked on and improved? Yeah, I think so. But at this point, we're just looking at the stain itself.
Thank you. I have one here for Dr. Ziegler. Are there any cost-benefit analysis studies performed regarding the application of Raman spec techniques in a typical forensic biolab?
Yeah, I don't know of any detailed studies that analyze cost-benefit analysis. There's obviously the upfront cost of the instrument, typical cost for that. For an instrument that I like, without naming it, is on the order of 25 or 30K. And then the consumables are largely the SIRS substrates, and those can be a range of costs as well. Homemade ones are obviously less expensive.
Here's another one for all the presenters, actually. Has your method been tested on age samples, or do you have plans to test them on age samples? And they're specifically, the question is specifically referring to samples that are older than a year.
So I can say that we've tested samples now that are going on 10 years old, and in terms of identifying proteins, there is some diminution of signal, but not much. And these samples were typically cotton, where neat body fluid or a mixture of body fluids were stained on them. Over time, we keep using the same ones over and over and over and look at loss of signal, which is too bad.
Well, I can comment on the aging and the SIRS work. For the most part, we haven't done any extensive studies of long-term aging. Certainly for periods of months of dried, aged body stain, the SIRS signature is robust. There's some indication, by the way, that for blood, that there may be small changes due to aging and further degradation of the protein that may be valuable in terms of judging, how old those stains are themselves. But for the most part, the features stay quite robust over the period of time that we've looked.
And this is Sarah. So in terms of the microRNAs, we haven't done an age study per se, but the way that our sample collection, we have a human subjects approval where we collect samples and then we store them dried at room temperature. And so I know that some of those samples are at least 1 to 2 years old, and that's a great idea for me to put on a student to pull that data, because I know that the data is very consistent for microRNA expression, regardless of how old the sample is, at least in that shorter time range.
And as far as messenger RNA testing of age samples, we have been able to do more than one sample over a year up to right now 4-year age samples. And we were able to see pretty good results with the messenger RNA targets, especially with the blood and the semen targets. But we are further testing some other samples to see how well the messenger RNA targets will do after a year of aging.
Awesome. Thank you all for the fantastic insight. Another question I have here, Dr. Williams, could non-human messenger RNA, or I'm sorry, microRNAs cause false positive serological tests or contaminations?
So that's a great question, because I did say that they are conserved across species. But what we found is that the expression levels, so one of the 6 microRNAs that we're testing may amplify with, say, monkey blood or dog blood or cat blood, but the 6 together will not give the same expression pattern. And so we have not really seen anything looking similar to what we see with the human body fluids.
Thank you. Thank you. Dr. Ziegler, for mass spec, how much sample is consumed, and does the sample consumption affect downstream processing like DNA?
Well, I can't answer that relative to mass spec, but for our platform, which is based on SIRS, the amount of material that we need for a SIRS signature is very small, which in other words, our samples are less than a microliter, or on the order of a microliter. And when we did try to obtain DNA profiling from that volume of, for example, blood, it was very difficult to get reliable results. So the main point I would make, even though we're using a procedure which is 50% acetic acid, is that the volume that's required for a SIRS result is very small, and too small from our experience anyway to get reliable DNA profile. The alternative is to use silver substrates. I didn't present any of that data there because the silver itself is more disruptive of the protein. We can just use water, which obviously doesn't interfere with DNA profiling of any sample. There, the sensitivity is pretty good. I mean, it gets down to, we can see dilutions of 10 to the 4 with silver reliably, and with gold, we do an order of magnitude better. But my point is that with just a water preparation in the silver substrate, that doesn't present any interference problems for DNA profiling subsequently at all.
Awesome. Thank you. I have another one for all the presenters. Could any of the methods presented today will they, or could they become, are they currently cost effective enough for possible adaptation by smaller, generally underfunded labs in more rural areas?
So I can begin, since we're at a public forensic laboratory. I don't think that is possible, at least for mass spectrometry, because of the investment in necessarily the cost of analysis per sample we calculated was less than $1, but that does not include the investment capital in equipment. At least at this point, I would imagine it would be local county or state labs that would be doing it. In New York City, we're large enough that, you know, we do our own, and we also aid local jurisdictions as well. But I don't think mass spectrometry would be something that a small laboratory would do. Having said that, I can say that if you have a toxicology unit, the mass specs that are used in toxicology could be used for body fluid analysis. So if you're large enough and you have a toxicology unit, then yes. If not, you would probably have to send them to a larger lab.
This is
Again,
Sarah.
I can comment. Go ahead.
Okay. So in terms of the microRNA analysis, absolutely. So we wanted to make things as streamlined as possible. And so that's why we're trying to get this to work with DNA extract. So that's what we're working on now, so that there doesn't have to be an additional extraction method. And then the analysis method is using qPCR, which even rural DNA labs have in their lab. So absolutely.
I'll again just comment on the cost of SIRS. it's on the order of, an instrument would be on the order of $30,000, let's say. So I have no idea where that falls with respect to, budgets in these laboratories. I guess it's an inexpensive car, something along those lines. The cost of the test, once the initial investment is made, is relatively minimal. So, I just, again, not knowing what the ballpark of a budget would be, that's the upfront cost, something on the order of $30K for CERS.
And this is Tiffany. I definitely think that the LAMP assay is something that could be used in smaller labs because it's an isothermal amplification method. You really only need a thermocycler or even a hot water bath to perform the reaction. And the detection method is using a camera. And so as long as you can keep the lighting consistent, there's really no high cost there because we've done this with a typical smartphone that most people have. So I definitely see it as something that could be used in smaller labs.
Awesome. That's great news. Thank you all for answering that question. I think that's on quite a few people's minds as we try to get further adoption of these type of technologies as they're emerging. We're actually going to head and wrap things up. It is the top of the time we have for today. So I'd like to go ahead and first thank our subject matter experts for taking the time out of their day to present the material for today's webinar. So thank you again, Dr. Siegel, Dr. Williams, Dr. Ziegler, and Ms. Lane for taking the time. Fantastic presentations, fantastic dialogue after the fact with answering the questions. And we really, really appreciate all that you were able to do for this webinar. So thank you. We'd also like to take this time to thank our audience for joining us today. These webinars would not be possible without your attendance and support. As some of you may noticed, there is an additional resources section available back on the webinar's main page. We're going to put a link to that page in the chat, and it's a great place to start for anyone interested in reading further into each of our presenters' research and findings. Before you leave today's webinar, we'd also like to ask you to participate in a short survey, which you can also locate back on the main page of this webinar. You can just click on that word survey, and it'll bring up our quick customer satisfaction survey for today's event. Your feedback about our webinars is vital information that we use to provide you the best webinar experience and adjust the content in the way we disseminate the information to our audience. One more thing to draw your attention to, you should see we are putting links in the chat to all our social media pages, which include Facebook, LinkedIn, and Twitter, a link to our website, forensiccoe.org, and a link to our podcast episodes. So make sure you are following us on all social media and subscribe to our newsletter to stay up to date with everything the FTCOE is doing in the forensic science community. Just wanted to say thank you again for attending. We hope everybody has a great rest of your day.